Many of us are already aware that obesity holds serious negative effects on human health, and that metabolism and appropriate fat storage is essential for maintaining good health. Yet how is it possible to begin to reduce obesity within the human population? A study has shown that nematodes may hold the agent for treating such disorders as obesity. When screening for new pharmacological agents, there is always a trade-off between the ease and speed and whether the chance that agent will work in humans. The nematode worm C. elegans is the ideal model organism for various studies due to the cost efficiency and the basic genetics which are able to be manipulated. Pharmacological agents that restrain energy balance within the context of whole organisms may be the link for understanding and the treatment of metabolic disorders. George Lemieux and fellow scientists approached this challenge by discovering which compounds regulated fat storage in groups of wild-type C. elegans in a basal metabolic state with 3,200 different compounds. Just like mammals, the fat in C. elegans is derived from dietary uptake and the metabolic regulatory pathways. Lemieux developed a chemical screen for new compounds that altered fat accumulation, and the reports showed that several molecules that either elevate or depress fat storage by combining compound treatment with systematic gene inactivation. Lemieux also decided on using null red dye, which has been extensively used in several experiments to identify pathways involved in fat metabolism. Null red staining of C. elegans intestinal organelles have a vast dynamic range whilst being sensitive to numerous genetic and environmental manipulations expected to alter fat content. Animals treated with AGRC1020 showed decreased staining as predicted, and these experiments indicated that C. elegans fat storage can modulate at the level of primary signal transduction. The nematodes were scored for growth and null red's fluorescent levels. They found that the selected compounds 1% lowered and 0.4% elevated, as Lemieux discovered certain compounds reduced the null red staining, whereas others increased. As the scientists were able to identify the compounds which altered fat metabolism in the nematode, they began to examine the effects of transcriptional expression, and once the findings were completed, it indicated that the screening strategy succeeded in identifying compounds within the fat levels. Further investigation continued to observe changes in fat by examining growth and feeding rates. Compounds that induce a low-fat phenotype cause either normal or higher feeding rates, and vice versa. Just as small molecules have helped drive understanding of many biological pathways, there is a need for pharmacological tools to examine complex process of whole organisms. Nematodes have proven to be compatible with the demands of chemical screening, as the findings suggest that C. elegans fat is flexible to large-scale screenings and sufficient to identify new compounds that modulate fat. Lemieux has reported in additional findings that C. elegans vital null red staining provides an effective strategy for identifying small molecules that disturb fat storage.